Great. Thank you for that introduction, and thanks for the invitation to speak. Can you see my screen? Good, and you can hear me.
Excellent.
Oh, good. Okay, so yeah, I'm just going to jump in. The title here is Biocontrol of Invasive Frag in Canada. I'm going to talk more generally about Canadian biocontrol programs as well. I'd just like to acknowledge my co-authors here, Mike McTavish, Ian Jones, and Sandy Smith at the University of Toronto. And later on in some of the slides, I acknowledge my Ag Canada colleagues who are, it's not all me doing the stuff that's on the tables that I'm going to show. Okay, so we'll just jump in. There we go. Okay, so I'm going to give you some background about biocontrol, how we do it in Canada, the approach we take, then specifically talk about Phragmites biocontrol agents. Then I'm going to talk about the operational release protocol that we've been developing to get them out there for frag, and overview of where we are with that Phragmites release program. So just so that we're all on the same page, biological control, we're talking about the use of live organisms, in this case insects, to suppress and introduce pests, in this case a weed. Why do we do that? Well, the weeds are introduced and there's no predators for them. So what we're trying to do in this program, and many programs in the States, reuniting the weed with the biocontrol agents to try and restore some sort of level of ecological balance. We're not trying to eradicate the weed. We're just trying to bring it down to some sort of threshold that we can tolerate. It's cost effective at the scale of the problem. You're all familiar with how big a problem invasive species are, and often they're beyond anything we can do with the management tools that we have. Biocontrol is another tool that can complement the ones that are out there. It's self-sustaining. The insects track the host plant and move with it. We'll find new host plant populations. It has a relatively low environmental impact. The only agents that are released are agents that are host specific to the target weed. That's what we spend an awful lot of time doing, making sure before they even get out there that the weed biocontrol agents are specific and only will attack the target. And finally, it's another tool for integrated weed management. I say it's another tool. It's not a panacea that's going to solve all our problems. It's just another tool in the toolbox that hopefully can help. So I want to highlight here, when you got a situation like this with frag, biocontrol is often the option of last resort in many of the programs that we do. We try, a new weed comes in, we try and eradicate it, stop it from spreading. We try all kinds of other tactics, and then at the end we say, well, let's try biocontrol. When you have an invasion like this, you have an option to do options. You can do nothing. You can use herbicide, cultural control, biocontrol, some sort of integration of these things. And I want to highlight that all of those options have some level of target non-target impact and non-target risk. There's no such thing as zero risk. And doing nothing and letting the invasive spread is going to have a lot of impacts on native species, T&E species that we're all concerned about. Biocontrol is not a zero risk proposition, but we work very, very hard to make sure that the agents are specific, will only attach to the target, and that there is a very low risk. Those are the agents that get approved for release. So this is what we're trying to do. Our goal, we have a weed. The weed's at a high population, the blue line here. We introduce the weevil populations increase, weed populations go down, and we get to some sort of equilibrium down here that's a lot more tolerable. Notice that the range of time here is 13 years or 15 years. It's a slow process. Most of the weeds that we're dealing with have a big head start, might have been here for 100 years, so we're not going to control them in 10 years or short time periods across the whole landscape. So this is a long game. We're trying to get something reestablished. Biocontrol has a long history in Canada. There's a series of books published with CABI going back to 1910. There's one in preparation now, should be coming out next year, detailing the weed biocontrol, insect biocontrol, and forestry programs in Canada across the years. In terms of weeds, the first agent released was for St. John's wort in 1951. And since that time, there's been 86 foreign insects released against 34 weeds. So it's been going on for quite a while. How do we do it? These are sort of four big areas that are out there in terms of activities. horizon scanning, exploration, risk assessment, release, post-release. And then these are specific steps or stages that we go through in deciding to have a biocontrol program. So starting with step one, some new invasive comes on the horizon, a new plant comes in, and one of the first questions we're asking is what's its impact, its ecological impact, its economic impact, is a biological control program justified, both the research and the risks associated. If we proceed through that lit review, and that's an ongoing process, and decide, okay, it's worth doing, we move to overseas exploration in the country of origin, wherever this thing came from, looking for potential specific biocontrol agents. Assuming that we find some that look promising, then we move to biology and host range studies, again, where we're testing those plants or the insects against non-target species to make sure it only attacks the target weed. If after, and that usually takes quite a while, as I'm going to show you in a minute, if it looks good, we have a specific agent, the testing is completed, we petition to regulators, both in Canada and usually jointly with the US, for the release of the agent. If it's approved by the regulators, we then move to research on rearing, how to get it out there. It's not a trivial question about how to produce enough of these, and then how do you establish them in the field? Step 6, stage 6 is once you get establishment, is what kind of impact is it having? And finally, if it's having an impact, there's redistribution and long-term assessment. And that long-term assessment is in a key step, important step that always is hard to do because you're talking about a long-term window of following up on these and keeping track of what's going on. So this is a really ugly table. I just want you to look at the columns on this stage. This column in yellow, those are the 7 stages that I just talked about. And then we have in this column in blue, those or aqua, those are the activities in each of those stages. And then over here, we have the targets. So what we have here is weeds that are being targeted and what stage they're at in Canada. And you can see there's quite a number of them, and they're at a variety of different stages. Highlighting here that all of this work is done with colleagues in Agriculture Canada. So I'm not the lead on all of these by any stretch, and there's other team members involved. I just also want to highlight that an agent, a weed can have agents at multiple stages. For example, with garlic mustard here, we have a seed weevil that's close to being petitioned for release. We have a stem root weevil that has been approved for release in Canada and is under study under field release and propagation. Similarly with yellow toadflax, it's farther along. This is work by Rose de Klerk Float. Very successful agent Messinus genthinus is in a long-term redistribution and monitoring stage, whereas Rhinusa pilosa, she's got it established at sites. It's looking very promising, and they're basically documenting what is it doing at those initial sites and what characteristics does it need to be spread to new sites. Just some new agents that have received approval in Canada, one for Russian olive, flowering rush, oxeye daisy. These are moving into that field release and propagation research stage. So I said I was going to talk about common reed. We'll move on to that. In this case, we have 2 moths, Arcanera neurica and Lanesia geminipuncta, which has previously been called Arcanera. So that may have been the name that you've seen it under. So moving over to that stages again, the Phragmites biocontrol program, and this was started by a lot of colleagues in the United States and overseas in 1998. Biocontrol was proposed, and there was that initial exploration for agents, period where they were looking at all the candidate agents that were available. There was a long list. By 2007, they narrowed in on these 2, neurica and geminipuncta, that were merited additional host range testing work. What I mean by host range testing, it uses this, what's called the centrifugal phylogenetic method, where you start looking at what it does on the invasive Phragmites. You also look at plants or representative species, others in the Phragmites genus. Then you look at plants that are in the same family as Phragmites, representative species, and it just continues out in terms of the proximity taxonomic proximity to Phragmites. You also include a list of species that are crops, economic crops and ornamentals for screening. So that screening in the case of Phragmites took 11 years or longer, basically 13 years, till we got to the point in 2018 where a joint petition was submitted for release of these two moths in Canada and the United States. The petition was approved in Canada in 2019, and that's where we are in the present, and what I'm going to talk about now with Canadian release studies. So these are the two moths that we're talking about. They're not sort of charismatic megafauna. They don't really show up that well, but they're very effective. So the moths aren't really the thing that does the damage. It's the larvae mining the shoots that cause stem wilt and mortality of the plants. And what we're trying to do, again, restore that ecological balance by making the Phragmites less able to compete. We're trying to kill stems as well, but we're trying to make the stands more suitable for native species to compete and get back to something more manageable. Okay, so just the biology of the moths. The larvae are hatching in spring, feed on the shoots for about 6 weeks. They then pupate for about 26 to 40 days. Adults fly for about 2 weeks. The Neurica has one instar less than the Geminipuncta, so they're out first, and then the Geminipuncta come out and fly. Both insects were screened and released with the idea that they're complementary. They don't overlap that much in their feeding, and we're trying to get a double hit, basically, on the target. The females will then lay about 150 eggs under the leaf sheath. These are the eggs right here, and then those eggs overwinter. You only get a single generation of each insect per year. So as I said, we're at stage 5 with the biocontrol agents for Phrag. And the program since 2019 has really focused on developing core methods on how to rear it, including artificial diets. What's the best way to release it? What's the best stage to release? And then how do you monitor effectively following those releases? And together, those come together in what we're calling an operational protocol for biocontrol or invasive Phrag that eventually we would hope to transfer to land managers, particularly the release methods and the monitoring. So I've broken that down here in terms of when those happen over the season. Today, I'm just really going to focus on the release and the monitoring part for interest of time. I'm not going to talk about the rearing work. So release work. We can release, we've done work releasing eggs, a little bit later larvae, and also pupae and moths. Generally, we're trying to get the material out there, tied to the phenology of the host plant. This is particularly important for the eggs when we want to get them out there at a time when the plant's out of the ground and there's something to attack. There's trade-offs depending on whatever release method you use in terms of efficacy and efficiency. By efficacy, I mean how many survive, and efficiency, how easy is it to do? And there's definitely trade-offs. So in terms of eggs, this is what we do. It happens sort of early in May. These are the eggs here that have been harvested from rearing. The good thing about it is that you can handle relatively large numbers. They go into this smaller cup inside a larger cup that has a mesh screen in the bottom of it. That cup is then closed up, and they can drop out the bottom into the Phragmites. The reason we've gone with this is we tried earlier looking at releases with eggs just stuck to cards, and there was very heavy predation, partially because they were out for quite a while. But this method provides some protection from weather and predators. The other next method is releasing larvae. So in this case, we let those eggs hatch into these larvae crawling around in the petri dish. And then each larvae is placed individually into a Phragmites stem. The stems are then put in foam blocks sitting here, and then those blocks are transported out into the stem and placed into the stand. This is the good thing about this is it's a lot more work putting them into these individual stems and all the transportation and such, but you do get better survivorship. You can also have the larvae in the stems, put them in there in the lab, and then hold those blocks for up to a week before the larvae are going to emerge out of those stems and look for a new one. This is how the insect really causes its damage, is that it moves between 4 and 5 stems or more, feeding as it goes during its life cycle. So once they're out in the plot like this, they're gonna come out of those stems and then seek out an appropriate new stem to bore in, and generally you get very good survivorship. We've also tried releases of pupae and moths. In this case, the pupae are in the vermiculite at the bottom of these containers here. The moths hatch out. They're very difficult to see. There's one right there, and it crawls up these mesh and then flies away. We did this initially because there was a time when we had pupae available to do it. It's a lot of work, partially because you have to rear all the way through to pupae and moths. But it was more whether or not it would work. Short answer is yes, and much to our surprise, I mean, basically you're putting these moths out in a frag stand in late June. They're going to hatch out as a moth and fly away probably, but actually they will stay around. You will get damage the next year within quite close to the release of where you put the release device, and so it is a method that works. But you have to do a lot of work to get them out there. Okay, I'm going to move on to the monitoring. In this case, we're monitoring in June, looking at larval damage. This is in the first year, so basically after the release. So here's a case where we have a release canister with eggs in it, and then the sample area around that is a 0.65 meter hula hoop. And what we do is evaluate all the stems in that area around the release. So this is what we're looking for when we're evaluating boreholes where the larvae have gone into the stem or out of the stem. We're also looking for stem wilt, which is an indication that the stem has something in it and it's died off. It's very diagnostic if you have these short little stems that are browning off, basically. You can also get branching on the stems, and if you're looking again, we're looking for pupal windows, because just before they pupate, they chew almost through the stem, so that when they come out as an adult, they've got a place to go. So those are the things that we're looking for in terms of larval damage. And then in the fall, and within that same year, we also do an evaluation, again, within that same sort of area, of the rammet and stand characteristics. And what we're looking for there are things like stem density, stem height, and panicle formation. And it's all of the stems are evaluated. This is the sort of data that you can get from this. In here on this bar, we have undamaged stems and the percentage or number. That 71% formed a panicle, whereas where there was feigning damage, only 8% formed a panicle. So right away, even in that first year, you're able to see impact on those stems that they're feeding on. Then in the second year or subsequent years following up, you have to come up with a different way of monitoring, because basically in the first year, the moths have emerged around those hula hoops, basically, and flown off and spread through the Phragmites patch in which you're releasing. So the way we do that is with a 1 meter by 4 meter transect, or where we walk along this transect, counting all stems that we see with damage on them. And up to 1 meter into the patch. And we basically played with this in terms of adjusting the size and trade off on the amount of time it takes to really, when you've got a huge patch, those of you that worked in frag know how difficult it is. And this comes up with a pretty good capture of what's going on in the field. So where are we with the release program? Well, since 2019, these are where the releases are located in Ontario. Starting then, the total number of releases are the bars here, and we're up to 30. And basically the number of individuals, either eggs, larvae, or pupae that have been released is just under 25,000 that we've gotten out over the last 4 years. And what we're seeing in terms of those monitoring that I talked about is in year one, the year that the insects go out, there's damage at up to 92% of those 30 sites. So in other words, this is a release site. When we go back to monitor, we're able to find evidence that there was feeding and something happened. And it ranges from between 0.2 stems per damage per meter squared up to 33.5 across those 30 sites. So very encouraging for first year data. Even more encouraging is in subsequent years, and we're now up to year 3 for some of the sites that we're monitoring. There was damage at up to 82% of the sites in year 2 and 86% of the sites in year 3. And the range of damage, and this is across those longer quadrats where we're sampling throughout the patch, more or less. We're still getting ranges of damage from 0.2 to 5.2 stems damage per meter squared. So it looks quite encouraging in terms of getting establishment of these insects. So just to sum up where we're at, the program started in 2019. We have an operational protocol that includes rearing, monitoring, and release methods for invasive Phragmites. We've released 24,000 insects at 30 sites. 3 years of monitoring data, we're confirming successful reproduction and overwintering of the insect at the majority of those release sites, which is very positive. And working on a lot of different biocontrol systems, seeing this sort of relatively rapid establishment at almost all of the sites is not that characteristic and very exciting. And so we're moving towards population establishment and nurse sites for redistribution. So the next steps, I just mentioned the nurse types. This, again, the map of the release location. We're continuing to monitor those existing release sites. We will be establishing new release sites associated with specific experimental questions to do with wet and dry locations, to do with pheromone monitoring adult moths with pheromones, and also, again, looking at what sort of stems do they prefer and in what sort of location within the release patch. But our hope is basically that these sites are going to take off and we'll be able to use them as nurse sites where we can recollect for them so we don't have to rear in the lab and redistribute easily in the field. So with that in mind, we've sort of moved on to, well, okay, how do we move these things now that we've got populations that we think are established? So we're now going out in sites where there's relatively high densities, or the highest densities we've seen, sampling stems in there and seeing how many eggs do we get. In this case, one site dissected 40 stems from the interior and found 128 eggs, which basically means 3 eggs per stem, which means you can then go out, and that's not a bad unit of moving that number of stems to a new location. And that's the sort of research questions that we're trying to address now. How much and where do you sample to get those stems from to move them to new locations? And this is just an example of the sort of data you get, the height of the stem from the ground, the number of eggs found. So most of the eggs are found in the lower 100 or lower meter of the stem, or so under leaf sheath there. And this is just a straight operational thing. It means we don't have to cut the whole stem. As you know, Phrag is really big. We can cut the bottom 100, take that back to the lab, and we're getting a reasonable snapshot of what's out there. We're also going to get enough eggs when we go to do redistribution. So that's the next step, or that's another of the key research questions that we're trying to address. So I'd just like to acknowledge a whole bunch of collaborators and supporters in terms of finances. And I'll stop there and happy to take any questions if there are any.